Hello, hi, my name is Faik Gökalp. I am from Kırıkkale University. My field of study is biochemistry. In particular, I am researching the effects of various active ingredients on diseases. The title of the statement I will present here today is a theoretical research on the anticancer properties of folic acid. It is a man-made version of folate, naturally occurring B vitamin found in many foods. Our body can't make folate, so it must be obtained through the dietary intake. Although the words folate and folic acid are often used interchangeably, these vitamins are distinct. Synthesized folic acid differs structurally from folate and has a slightly different biological effect in the body. That said, both are considered to contribute to an adequate dietary intake. Folate is found in a number of plant animal foods, including spinach, kale, broccoli, avocado, citrus fruits, eggs, and beef liver. Let's see these foods. Okay. Folic acid is a water-soluble vitamin and important for biological systems. It is a highly stable vitamin that dissolves in water. It is in the form of light orange in crystalline form. The most important metabolic function of folic acid is the transfer of single carbon to the related metabolites and the synthesis of purines and pyrimidines, which are necessary for nucleic acids. Folate deficiency causes impairment of DNA synthesis. Let's see the structure of folic acids in the picture. Okay. Yes, the structure of folate, it has three parts. You see the structure of folate in the picture in the screen, on the screen. The hydrofolate reductase inhibitors are important drugs for anti-cancer. Folate deficiency can lead to megaloblastic anemia and cancer. The crystal structure of alpha amylase, PDBID, B1HNA, downloaded from Protein Data Bank. In the study, the inhibition effects of folic acid will be investigated theoretically with the values obtained from the docking scores determined by the docking method for the anti-cancer properties. BAL27862 has an interesting chemical scaffold bearing a benzimidazole functional group. BAL27862 is a new microtubule destabilizing drug undergoing phase 1 clinical evaluation as prodrug. BAL 101553. This agent is a potent inhibitor of tumor cell growth and has shown a promising activity profile in clinically relevant strains of human cancer models resistant to microtubule targeting. BAL 101553, called as lisavanbulin, the lesion prodrug of BAL 27862, called as avambulin, has been found to exhibit broad anti-proliferative activity in human models. Lisavanbulin, prodrug of avambulin, is a new tumor checkpoint controller that promotes cell death by modulating the checkpoint in the mechanism. Lisavanbulin, the prodrug of the microtubule destabilizer avambulin, has been shown to exhibit antitumor activities when previously administered as a short-term infusion, but is associated with toxicity in its use. BAL 101553, lisavanbulin, is a novel synthetic molecule exhibiting promising anti-tumor activity in non-clinical studies. Avambulin has the property of being solubility lesion prodrug. Let's see the results and discussion. The interaction of folic acid with various cancer cells is given in Table 1 by comparing the standard BAL 27862 and BAL 101553 code docking scores. The interaction docking scores of folic acid with various cancer cells and standard BAL 27862 and BAL 101553 is given in Table 1. When we look at the Table 1, we see the ligands and receptors. Ligands, folic acids, and the other two reference substance for as a drug. The other receptors are 1, 2, 3, 4 receptors, cancer pathway. First one receptor was overexpressed in 85% and 50% of human colorectal carcinomas and adenomas for 3L and 1 downloaded from Protein Databank. The other PDB-ID downloaded from Protein Databank found in the literature for 3QUM, 1HNA, 1DQ8, simvastatin, A3-hydroxy 3-methyl CoA, Predictas, called as HMGCR inhibitor, a drug commonly used to inhibit cholesterol synthesis, is a compound with anti-tumorigenic activity. Tumor-specific HMG CoA predictors, called as HMGCR expression and good prognosis have been previously observed to be effective in breast and ovarian cancer. Given the effects of HMGCR as a predictor of response to neoadjuvant or adjuvant statin therapy in colorectal cancer, further studies are required. Look at the results in the table 1. It appears that standard ligands with low energy binding, docking scores, and folic acid interact with amino acid residues in cancer cells and cause inhibition. Let's see the result of ligands and receptor affection. For the first one, the inhibition effect from large to small, BAL 101553, folic acid, and BAL 27892. For the second one, the human prostate, and the inhibition effect from the ligand and receptor from large to small, BAL 101553, folic acid BAL 27892, 862. For the third one, pancreatic alpha amylase, the inhibition effect from large to small, BAL 27862, BAL 101553, folic acid. For the last one, for human, HMG and with the ligand with the receptors, the inhibition effect from large to small, folic acid BAL 101553, BAL 27862. The inhibition effect of folic acid to HMG and CoA is given in figure 1. We see the figure of it on the screen. As a result of values in this table, folic acid appears to be very effective in interacting and inhibiting amino acid residues in the specific cancer pathway, as well as reference drugs use it as standard. It can be concluded that this interaction is polar, hydrophobic, and hydrogen bond interaction through functional groups, electrostatic force, hydrogen bonding, hydrophobic interaction, non-covalent interaction such as ionic, hydrophobic, and hydrogen bonds. If charges are present, occur through the functional groups that interact with the binding site substrate of an enzyme. Let's see the conclusion. In the study, it is understood from the values of the docking energy scores that folic acid, which has an important function in single carbon transfer and nucleic acid synthesis, also has an important inhibition effect on various mechanisms on the cancer pathway. This theoretical study is important in terms of preventing the loss of time and substance, determining the active substance in advance, and directing the experimental studies. Yes, let's see the reference to this study, research this study. And thank you for listening to me